Right, you will put in and ask this. Cytokines is a bit dry subject, so it will have so many components like definition, properties of cytokines, and nomenclature, classification, cytokine receptors, cytokine signaling, cytokine antagonists, and T helper cell 1 and 2, and cytokine related diseases, cytokine therapy, and cytokine hematopoiesis. So these are the components which we study. It's a common question that is asked, but it's a bit tough subject. So the development of an effective immune response involves lymphoid cells, inflammatory cells, and hematopoietic cells. The complex interactions among these cells are mediated by a group of proteins collectively designated cytokines to denote their role in cell-to-cell communication. The cytokines, immunocytokines, in Greek, cyto means cell, kinos means movement, and low molecular weight regulatory proteins. They are the low molecular weight regulatory proteins, or they are the glycoproteins secreted by white blood cells and various other cells in the body that responds to a number of stimuli, that is due to infection. The properties of cytokines they bind to specific receptors on the membrane or target cells and triggering the signal transduction, touching with each other or communicating with each other. The pathways that ultimately alter gene expression in target cells. So the cytokines are their fully assembled receptors exhibit a very high affinity for each other and deliver intracellular signals. The cytokines and their receptors exhibit a very high affinity for each other, and with the dissociation constant ranging from 10 to the power of 10 to 10 to the power of 12 microns. Because their affinities are so high, the cytokines can mediate biological effects at premolar concentrations. Inducing stimulus, so that is due to injury or any other inflammation, will act. The cytokine gene that gets stimulated, the cytokine producing cell, that is the cell that is mostly the T cells, and the cytokines are released into circulation. It goes and attaches to the target cell or any other cell. This is the biological effect of cytokine, which will go and become adherent to the other cells. Like infection, bacteria will go and attach to the bacteria. a particular cytokine and may bind to receptors on the membrane of the same cell that secreted it, or exerting the autocrine action, and it may bind to receptors on a target cell in a close proximity to the producer cell, exerting a paracrine action. So in a few cases, and it may bind to target cells in distant parts of the body, exerting a endocrine action. So this is an autocrine action where the cells, the cytokines will become adherent to the cell of its own origin. The paracrine, and they will go to a nearby cell and get adherent to the nearby cell, and it goes to the circulation, and it also goes to a distant cell where there is injury or inflammation in the body, and it will go and becomes adherent to that particular target organ. The cytokines exhibit the attributes of pleiotropy, redundancy, synergy, antagonism, cascade induction, and which permit them to regulate cellular activity in a coordinated interactive way. So this is the target cell, and where this is the target cell, and from which the B cells and T lymphocytes. The B cell will go into the thymus and becomes a thymocyte, which becomes more active to produce interleukins, interferons, etc., and it will be transformed, and the activated T cells will be transformed into mast cells, and so these are all the cells which will produce interleukin 2, interleukin 4, 3, like that. The interleukins will be produced by different cells. For example, the nomenclature, you see now, the interleukins that act as mediators between leukocytes, and the vast majority of these are produced by T helper cells, and lymphokines produced by lymphocytes, and monokines produced exclusively by monocytes, and interferons involved in an antiviral response. So the colony stimulating factors support the growth of blood cells. The chemokines mediate chemo attraction, that is chemotaxis, between the two cells. The cytokine families, you have hematopoietin family, interferon family, chemokine family, the tumor necrosis factor family. So in this you have the cytokines have been classified on the basis of their biological responses into pro or anti-inflammatory cytokines, depending on their effects on immunocytes. The major cytokines include lymphokines and interleukins and monokines and interleukins, interferons, and the colony stimulating factors, and the tumor necrosis factor alpha and beta. They all come under cytokine group. So the type 1 and 2. Type 1 is cytokines produced by T helper cell 1 and T interleukin 2 and interferon gamma and interleukin 12 and in tumor necrosis factor and type 2 cytokines are those produced by T helper cells include interleukin 4, interleukin 5, interleukin 6 and interleukin 13. These are all of these cytokines. The mediators of natural immunity or tumor necrosis factor alpha, interleukin 1, interleukin 10, 12 and interferon. Type 1 interferon is interferon alpha and interferon beta, interferon gamma and chemokines. The mediators of adaptive immunity or interleukin 2, interleukin 4, interleukin 5 and tumor growth factor beta and interleukin 10 and interferon gamma. So there are all the things which are called pro-inflammators, reactions which take place from usually the cytokine promotion activation that usually occurs, differentiation and proliferation or cell death or T cells of cell death of T cells, that is, and B cells, macrophages, dendritic cells and natural killer cells and other leukocytes. And usually it starts as whenever there is inflammation, the stem cell from which the adaptive immunity gets stimulated and which stimulates the macrophages. The macrophages are formed from T helper cells. So the T helper cells will be transformed into macrophages. These macrophages are also come from hematopoiesis from the bone marrow, from stem cells. And the macrophages which will synthesize the cytokine promotion by the help of T helper cell differentiation. So this is an inflammatory reaction that occurs due to cytokines. The cytokine act in an antigen non-specific manner. The non-specificity of cytokines confirms with establishing a specificity of immune system. How does this work? Careful expression of the receptors for those cytokines on specific cells, and then receptors are often only expressed after exposure to antigen. The cytokine receptors fall within 5 families. The receptors for the various cytokines are quite diverse structurally, but almost all belong to one of and the 5 families of receptor proteins. The immunoglobulin superfamily receptors, superfamily receptors. That's class 1 cytokine receptors family, also known as the hematopoietin receptor family. Class 2 is cytokine receptor family, also known as the interferon receptor family, which produces interferon, alpha interferon, beta and gamma interferon. And you also have receptors, the chemokine receptor family. The receptor family, that is the immunoglobulin superfamily receptors, they are called ligands, interleukin 1 and main colony stimulating factor and c-kit, interleukin 19. So they are all interleukins, ligands, interleukin 2, 3, 4, 5, 6, 7, 9, 11, 12 protein and conserved cysteine and WS. The receptor family, there is a class 2 cytokine receptors, interferon, alpha, beta, gamma, interleukin 19, 10, 20, 22, 24, 26, 28, 29. The receptor family and with ligands, receive tumor necrosis factor receptors. You have C1, C2, C3, C1, C3, C2, C1, C3, C2. So they are all located on a cell. So they are only ligands, tumor necrosis factor, alpha, beta, and CD27L, CD3, 0L, 4, 0L, and outgrowth factor, and FAS ligands. They are all. The chemokine receptors are the G proteins. They are interleukin 8, antis, MIP1, PF4, CAF, NAP2, GM-CSF receptor subfamily common subunits like colony stimulating factor, interleukin 3 and 5. So the low affinity receptors, they're all interleukin 3, 5 and colony stimulating factor, which will be attached to the exterior of the membrane of the cell, and interiorly you have the beta subunit, alpha interferon. High affinity receptors, they have very high affinity towards the cytokines, and same signal from all three can be present. So they are all that's baseline. That line between the center is the basement membrane, and interleukins outside the membrane and inside the membrane, or interferons outside cytokine interleukins. Interleukin 2 or is the most thoroughly studied cytokine receptor, expressed by T cells for proliferation, called CD25 surface marker in mature T cells. Complete receptor have 3 subunits. The intermediate affinity interleukin 2 or the high affinity interleukin 2 or over the basement membrane, low affinity interleukin 2 or. The subunit composition, interleukin 2 or beta, 2 or gamma, they are all and interferons which are produced by T lymphocytes and macrophages, so which will help the cell expressed and the natural killer cells and resting T cells and which are low numbered and activated CD4 and CD8 and T cells activated B cells low numbers. Majority of the cytokines cytokine receptors are in class 1 and class 2. These cytokine receptors lack signaling motifs, and this was puzzling to researchers. The interferon gamma and was studied because of its ability to block viral replication and the other interferons. So, however, this interferon plays immunoregulatory role and can lead to understanding the process of signal introduction through cytokine receptors. The ligands which will bind the alpha subunit ligand binding causes dimerization of receptor, and JOX get activated receptor by phosphorylation of tyrosine residues on receptor and phosphorylation of JOX themselves starts DOG receptor. Phosphorylation of starts by JOX dimerized starts translocated to nucleus and then gene expression results. So the signaling pathway of cytokine system, due to dimerization, the cytokines will get attached to the cell membrane, and alpha and beta interferons and the JOG receptor, which is inside the cell surface, the cell membrane surface. The activation of the JOG receptor family, tyrosine kinase, phosphorylation of receptor, tyrosine phosphorylation of start by JOG kinase, and dimerization of start results in DNA transcription, specific gene that gets to transcription. For instance, example, constant irritation over a particular point will lead to all the transformations over the tissue and results in gene transcription and may form a squamous cell carcinoma over the skin. That is how, or any other malignancy for that matter. The cytokine antagonists also are present. The number of proteins can inhibit cytokine activity, can bind to receptor or can bind to cytokine, disabling it, and enzymatic cleavage of receptors and release of these can bind cytokines in the blood. Marker of chronic T cells activation, transplant rejection, and viruses have developed these strategies, and cytokine homologs, cytokine binding proteins, homologs of cytokine receptors, interference with intracellular signaling, interference with cytokine secretion results. So these are all some of the virus infections which will mimic like that of cytokines, the cytokine receptors, like lipopoxy virus, that is myxoma virus, and so it will produce interferon gamma and attaches to the receptor. And several pox viruses will produce interferon gamma receptor attachment and may help in producing malignancies, hematological, vaccinia, smallpox virus, soluble interleukin 1 beta receptor, or Epstein-Barr interleukin 10 homolog. The human herpes virus 8, interleukin 6 homolog, also homologs of chemokines MyP1 and then MyP2, and cytomegalovirus, and it will also produce 3 different chemokine receptor homologs, the one of which binds 3 different soluble chemokines and arantes and MCP1. So the T helper 1 and the T helper 2 cells, and which will the CD4 T helper cells exert most of helper function through secreted cytokines and two populations and based on secreted cytokines and cell-mediated functions and a delayed hypersensitivity activation of and T cells production of opsonization and promoting immunoglobulin G antibody to attach to the bacteria, viri, and may try to kill or swallow it down through the help of macrophages. That stimulates the eosinophil activation and provides a helper to B cells and promotes production of large amounts of immunoglobulin M and immunoglobulin E and supports the allergic reactions CD4 cells. And so the T helper cells do not show T helper 1 or T helper 2 profiles. So the cytokines of different types, that is, the cytokines profiles are cross-regulated. They promote growth of a subset that produces them and inhibit development and activity of other subset. Progression of some diseases depends on balance between T helper cells and T helper cell 1 and 2. The transcription factors, T-bet expression results in a T helper cell and T-bet suppresses T helper 2. GATA3 results in a T helper 2, GATA3 suppresses T helper 1. Interferon gamma regulates expression of T-bet STROT1. The interleukin 4 regulates expression of GATA3. So this is how the inflammatory process that takes place. Cytokine related diseases like SCID is a inflammatory subacute combined inflammatory diseases, genetic defect in cytokines and the receptors will be present. Defective receptor for interferon gamma, susceptible to mycobacterial infections. They are the people who are prone to develop infections and bacteria and septic shock, certain gram-negative bacteremia, and then some symptoms like drop in blood pressure, fever, diarrhea. They will have endotoxins bind TLRS and dendritic cells and macrophages and produce interleukin 1 and tumor necrosis factor also. Alpha and cytokine imbalance causes abnormal temperature, abnormal respiration, capillary leakage and tissue injury, organ failure. Neutralization of by monoclonal antibodies may be of great help in these individuals. And then you have bacterial toxic shock syndrome also can occur, and super antigens may bind simultaneously to class 2 MSC and T cell receptor activating T cell despite antigen specificity. Symptoms similar to that of sepsis they have, and abnormalities in the action of cytokines or receptors are also associated with certain types of cancer, Chagas disease, and caused by Trypanosoma cruzi, severe immunosuppression, and evidence that soluble factor produced by T. cruzi and leads to reduction in T cell interleukin 2 and CD25 receptor. The cytokine therapy is also to some extent available. It's a non-specific stimulation or inhibition of particular components of the immune system may sometimes be of benefit. The best results have been obtained with cytokines, and among these interferon alpha is the most widely used mainly for its antiviral properties, and the most striking clinical effect of a cytokine has been that of GCS colony stimulating factor, restoring bone marrow function to anti-cancer therapy. Then you have interferon beta 1 alpha and interferon beta 1b are used to treat and control multiple sclerosis and autoimmune disorder. So the interferon therapy is used in combination with chemotherapy and radiation as a treatment for many cancers. This treatment is most effective for treating hematological malignancy, leukemia and lymphomas, and including hairy cell leukemia, chronic myeloid leukemia, and nodular lymphoma, cutaneous T-cell lymphoma. Both hepatitis B and hepatitis C are treated with interferon alpha 1 and often in combination with other antiviral drugs. Administered infra and intranasally in very low doses, interferon is extensively used in Eastern Europe, Russia, as a method to prevent and treat viral respiratory diseases such as cold and flu. Interferon therapy causes immunosuppression, and in particular through neutropenia, and can result in some infections manifesting in a unnatural way. The discovery and that the beta chemokines and RANTES, they are the ligands, MRP, macrophage, inflammatory protein, suppress HIV-1 infection. So these are all the cytokine-based therapies and antiviral drugs, therapies which are used in like there are few of the drugs that are mostly used in antism. They interfere with the cytokines and relieve inflammation and pain. And even in malignancy, like where you use monoclonal antibodies like chronic myeloid leukemia, etc. The problem with cytokine therapies, or you see, the suppression of T helper cell proliferation and suppression of C cell activation and antibodies to interleukin 2 or an interleukin 2 or over the T cell, T helper cell. So leads to destruction of activated T helper cells. The problem is that and the effective dose levels and short half-life and potent biological response modifiers can cause unpredictable side effects and destruction of activated T helper cell that results because of the interleukins will go and attach to the target cell membrane in the extreme. The cytokine inhibitors the cytokine inhibitors can be used for severe or chronic inflammatory conditions like rheumatoid arthritis or any other inflammatory disorders, inflammatory bowel disorders, various ways of inhibiting tumor necrosis factor and interleukin 1 have proved valuable in rheumatoid arthritis and the controversial and septic shock and severe malaria also. The cyclosporin A and pentamidine. The cytokines play an essential role in hemopoiesis also. Hemopoiesis also it helps in producing the RBCs, especially in erythropoiesis. That is erythropoietin that has been tried and colony stimulating factor and thrombopoietin in liver and kidney diseases and to include platelet function, platelet production, and then this role the drugs that are tried in many immunological disorders. In summary, you see, the cytokines are low molecular weight proteins that are produced and secreted by a variety of cells. So, especially at biological activity of cytokines exhibit pleiotrophy and redundancy, synergy, antagonism, and cascade induction, and the families of cytokines, hematopoietin, interferons, chemokines, and tumor necrosis factor, cytokine receptors, immunoglobulin superfamily, and the class 1, class 2 tumor necrosis factor, and the chemokine receptors. The cytokine-induced multimerization of class 1 and class 2 receptors activate the JOT and the STROT signal, the ligands and transaction pathways, and that's how they act upon and are used in most of the chronic inflammatory disorders of all the systems, many a times and for all the systems, mostly used in rheumatology and hematology, and it is a great help in pulmonology with the inflammatory diseases of the lung and with idiopathic pulmonary fibrosis, etc. So this is of course, very dry subject. Should be read many times before the examination itself. And just before going to the exam, you can try once for Davidson. He has given two small. Within one page it comes in Davidson. That's enough to write for your standard. One page. Is it? అంతా రాయలేదండి.